All right, so welcome back today. We're going to finish up with cellular respiration today. So last time we talked about glycolysis, so the first step of harnessing ATP energy from glucose. And when we left off, we had looked at this process right here, going from glucose to 3 carbon molecules of pyruvate. But pyruvate has more energy to yield. There's 3 more carbons that we can oxidize. Oxygen is available, then pyruvate is able to enter the mitochondria, the powerhouse of the cell, and the enzymes of a circular sequence of chemical reactions called the Krebs cycle can be used to complete that oxidation. So if we talked about this part on the left last time, we're going to look at this part on the right taking place in the mitochondria today. Remember, just keep this in mind, the whole point of this is to make ATP energy. So it is oxygen. To the bloodstream, eventually to the lungs, and we breathe it out. So this kind of a mini step, it's like the link reaction sometimes it's called between glycolysis and the Krebs cycle. It's just a little further oxidation of pyruvate into this molecule called acetyl CoA. right? And through a step of chemical reactions, we're going to start to oxidize this citrate, going down to 5 and 4 carbons. And in doing so, we're removing carbon dioxide. Carbon dioxide is a sign that we're oxidizing something. So we're adding it to the cycle, oxidizing it, and what's left over is the 4 carbon substance that we can start out with again to begin the cycle again. This is actually called oxaloacetate. It's not something you have to remember. But so the cycle's regenerative. So by the end, we're left with a 4 carbon substance that can be turned back into citrate when we add some more acetyl CoA. Right? So that's just kind of the basics. Counting the carbons, understanding that we can recycle. If we add the electron carriers, where we're removing electrons in this process, it might look like something like this. When we remove carbon dioxide, that means we're oxidizing, we're breaking a bond, and so we can harvest some electrons in the form of NADH from it. And again, when we remove electrons, we can harvest some NADH and use it later. We haven't mentioned as much about FADH2. Just remember it does the same thing as NADH. It stores electrons, stores high energy electrons. And the last thing you might want to notice is we make some ATP directly in the process as well. So all of this comes out from one turn, one trip around this cycle. Each glucose molecule allows this cycle to run twice. Okay, so if we're thinking about in total what we make, 1, 2, but twice, 3, 4 molecules of carbon dioxide, 1, 2, 3, 4, 5, 6 molecules of NADH, 1, 2 molecules of FADH, and 1, 2 molecules of ATP. So that's pretty good. That's a lot of energy that we're getting out of this substance in the Krebs cycle. That's why it's so important, right? You don't need to memorize the different steps. Just like last time, what goes in? Pyruvate and acetyl, or eventually actually acetyl CoA. Okay, goes in. And what comes out? Carbon dioxide, NADH, ATP, and FADH2. Okay. You'll probably remember citrate. You don't need to remember oxaloacetate. You definitely don't need to remember anything in here. Similar to like what I said in our Zoom session yesterday, if you're doing this in medical school, you would have to memorize something like this. Actually, this is a simplified version of what you'd end up having to memorize. Um, but I'll leave that to if you want to go down that road in the future. So, when we fully oxidize glucose to carbon dioxide, right after the Krebs cycle, there's nothing left except carbon dioxide. We've ended up with 4 ATP in total. 2 from the Krebs cycle, all right, because we make 1 each turn and glucose turns it twice, and 2 in total from glycolysis, because we had to put 2 in to start glycolysis, and we got 4 out. So kind of what's the point? That doesn't seem like very much energy. 4 ATP? What's the point? Remember all of the NADH and the FADH2 that we got, right? We got 2 in total from glycolysis. We got 1 for each pyruvate molecule, so we got 2 NADH in the link reaction. We got 6 NADH from the Krebs cycle. We got 2 FADH in the Krebs cycle. Those are all stored energy in electrons, electron carriers. And we are at the final end going to make a whole ton of ATP by using that energy um, from those electrons stored in the electron carriers. So let's kind of look. From the Krebs cycle, we've net gained 2 ATP, 8 NADH, and 2 FADH2. We kind of talked about that already. So, if we only are getting 2 ATP, then why are we doing the Krebs cycle? And like we talked about, there's a value in these electron carriers, all right? From reducing those molecules to NADH and FADH2, we can use those electrons, and the electrons we stored earlier from glycolysis, in what's called the electron transport chain. All right. So in electron transport chain, you're going to see we're going to make a bunch of ATP, a lot more than 4 that we've done so far um, from directly making ATP by substrate level phosphorylation in the glycolysis and the Krebs cycle. The electron transport and FADH2 from where those electrons are liberated or freed in their reactions to the electron transport chain where they're going to be used. This is where they get dropped off. Passed from protein to protein in the chain because each protein is a little more electronegative than the last one. So they naturally travel one direction down the chain. And as by these proteins to transport hydrogen ions from the mitochondrial matrix to the intermembrane space, from the middle of the mitochondria to the inner membrane space. So as the electrons travel down the chain, they power these pumps that move hydrogen ions from in the mitochondrial matrix into the space. And when the electrons get to the last protein of the chain, they are finally passed to oxygen, a really electronegative element. And they combine with oxygen and some free hydrogen ions to make water. So this is the big reason that we need to breathe oxygen, is because it's the final, what we call electron acceptor in the electron transport chain. And if it wasn't there, these electrons couldn't flow and we couldn't make ATP. So here is another picture, okay, of them traveling down the chain, pumping those hydrogen ions, being pulled from a less electronegative complex protein to the next, all the way down to the most electronegative part, which is the oxygen at the end. So like we said, we can think of them flowing downhill, down a stairway, using up a little bit of energy each time to help that next protein in the complex, which is more electronegative than the last, pump hydrogen ions into that intermembrane space. So now we have a bunch of hydrogen ions in this intermembrane space. What are we going to do with them? All right. Well, there is a protein, an enzyme in that intermembrane space called ATP synthase, and it's going to synthesize ATP. And this protein is actually a channel protein where hydrogen ions can go through. So if we have a whole bunch of hydrogen ions in the intermembrane space, we have a high concentration there. And so they'll want to naturally diffuse through that ATP synthase. And this ATP synthase is like a generator. As hydrogen ions flow through ATP synthase into the mitochondrial matrix, its shape is going to phosphorylate ADP into ATP. So as hydrogen ions leak back through, this enzyme phosphorylates ADP into ATP. Okay, so as these go through, this sticks a phosphate onto ADP to make ATP. So it's kind of like a water wheel using the moving hydrogen ions to power the process of phosphorylation ADP to ATP. So we call this process of hydrogen ions diffusing back through ATP synthase chemiosmosis. Okay, and that's what really couples ATP synthase with the rest of the electron transport chain. We build up this gradient to power ATP synthase. That's the whole point. So if you think about it, all the food that you eat job is to power a proton pump. It's kind of a really weird way to think about it. All right. Um, Here is a picture of his name's Peter Mitchell, who first proposed the chemiosmotic hypothesis. And at the time, it was a revolutionary idea. No one was really sure how this worked, but he was the first one to kind of lay out what I just explained to you, and it changed a whole bunch of the ways that we understand about even the most basic biological processes. That is it for today, and we're going to go through this a whole bunch during our Zoom time tomorrow on Thursday.